Okay, so so what are we ended the last lecture um, talking about CMI effector responses and the fact that they, like we saw with complement, induce these porins, these perforins, these these holes, these protein-based polymeric holes in membranes, causing the membranes to lose their to cause the membranes to lose their integrity and eventually leading to cell death. Um, now, I thought I'd show you is that in our laboratory, we have developed um, a new technology that we call prosthetic antigen receptors, which we use to modify cells. And essentially, there are these nanorings, and let me just get this here. There are these nanorings here um, that can associate with the CD3 on T cells, and they can stick. They stick because they have high valency. They can stick to T cells, and then what they end up doing is they have an antibody, a binder that can target to the cancer cell, and therefore associate the T cell with the cancer cell, the CD8 cancer cell effector cell, causing activation and then cell killing. So we refer to these as prosthetic antigen receptors, and they're these nanorings are able to initiate this targeting. Um, so, and if you were to look at, shoot, if we were to look at cells, here's some cells. These cells here that are um, stained in red are glioma cells. These cells up here, which are the smaller cells, which are not stained, are breast cancer cells. And these little guys here are T cells moving around. These are what the T cells look like. Here's a cell that's dividing. And these T cells, the breast cancer cells, have an antigen on them that the glioma cells don't have. Now, the T cells can't recognize uh, uh, the glioma cells or the breast cancer cells, so they're unable to really kill them. They don't really stay around long enough to kill them, to kill those cells. Now, if we functionalize those cells, I don't know what this is here, but uh, if we look at these cells down here at the bottom, we functionalize our T cells with the nanorings. The nanorings, the T cells now are able to kill these breast cancer cells because they have the antigen on them. And you can tell they're killing because they collapse. The cells collapse. If we look up here, you can see these T cells here are all over this cell that just divided, and the cell is dead. And these cells are dead down here. And this is a hallmark you can see here of these cells. Um, they are necrotic, they have collapsed, and the T cells are gone. And essentially, I like this part here. You'll see it here in a second. If you watch this cell, the cells just would explode like that. And essentially what happens is the T cells are functionalized and now able to go off and kill the specific cells which have the EBCAM antigen on it. But the glioma cells are not killed, so you get specific specificity. So this is exactly what happens when a T cell, activated T cell, recognizes an infected cell and kills it. So I just wanted to show you this as an example of how T cells actually kill and can kill a target cell. So if we look at CMI effector response, again, just to look, here's a healthy cell. Once the cell has undergone apoptosis and interaction with the T cell, you see the membrane destroyed, the chromatin is being destroyed into these pieces. You see this chromatin over here is pretty is normal. It's big and normal, where it's all broken up here. And then in late apoptosis, the cells are just these bodies with pieces of the nucleus, and the membrane is all destroyed. Now, if we get back, we now remember what is the relationship between CMI and AMI response. We looked at a series of cells that are really dependent on the T cells. The T cells are the dependent actors that control this whole response. So if we look in this middle, we talked first about T helper cells, for example, Th1 cells. And the Th1 cells can go on and lead to activation, and then initiation of the CMI response. Th2 cells will engage B cells, and then lead to antibody production. So that's CMI, and so those are the two. So CD4s are involved in both CMI and AMI. And then CD8 positive cells are your cytotoxic T cells, which lead then to destruction of virus-infected or cancer cells themselves. And so in all this response, we have these are the sort of key varieties of T cells. Now it turns out that there's several different types of T cells, and we'll come back to that in a second, because there's one important other T cell I want to introduce to you. But if we look at the whole framework here of the AMI and CMI response, what we see is, so what we see here is on this side that we have the AMI response, right? We have this AMI response is here. Now, when a Th0 cell produces also Th1 cells, then you get the CMI response, which is here. So that's what we just talked about. So you can see this interaction, the central interaction here. Let's get the color here. You can see this key interaction here where you get the antigen presenting cell and the Th0 cell, and whichever way it goes, either through IL-2, by the influence of IL-12 or IL-4, gives you the Th1 or Th2 cells, and that influences whether the differentiation of the T helper cells goes down the pathway of a antibody producing response or down the pathway of a T killer cell, cytotoxic T cell killer response. And they can be both. You can have both occurring. For example, a Th0 cell could, if it's an effective cell, can go the way of Th1 cell. If it interacts with antigen processing cells that are presenting peptide that will initiate from soluble antigens, it could induce an IL-4 response and therefore produce antibody. And so typically what you have for a virus actually is you don't have just one of these responses, but you have both of those responses, in which the CMI response over here gets rid of the infected cells, and the AMI response here attempts to get rid of any of the virus that's essentially in circulation, to try to remove that. So you can see here how this all kind of fits together, and you can see centrally in here the T cells are the big players. These T cells, the big players that guide and control this function. If their interactions with the various cells are not optimal or they're not activated, then the processes won't go forward, and therefore there's a sort of there's control mechanisms that are guided by the T cells to govern whether you go forward or whether you don't go forward in producing an immune response. Now, there's one other type of cell that I want to talk about, and this is important because it's important for inflammation. So this cell is our Th17 cells. And so when we have a Th0 cell, what we've learned is that if it's in the presence of IL-4, you'll get Th2 cells, and they'll go on and produce more IL-4, which produces more Th2 cells. If it's in the presence of IL-12, now these are due to the microenvironments within those cells, how the antigen processing cells are, what's happening there. IL-12, then you'll get Th1 cells, and they'll produce interferon gamma and IL-2, and this you'll have your CMI response. It's turned out that there was another antigen, another cytokine. Let me just see, IL-17. This is interleukin 17. It was recognized that this is a pro-inflammatory cytokine. It's about 155 amino acid protein, so a reasonable size glycoprotein, meaning it has glycosaccharides on it. It's recognized that this IL-17 induces antimicrobial peptides and many other inflammatory cytokines. It's a chemoattractant in that it attracts neutrophils into sites of inflammation, and therefore sites where there would be the need for there may be infection, or if there's an injury, the need for regeneration and protection against infection. And it's also was known that it was strongly associated with inflammatory disorders such as psoriasis, multiple sclerosis, rheumatoid arthritis, as well as others. Now, as more research was done, it was discovered that actually the IL-17 comes from a type of CD4, a CD4 positive T cell, T helper cell. And that is not the same as Th1 and Th2 cells. And because it produces IL-17, it's referred to as Th17. Now, this transformation of the Th0 cell in these sites of inflammation, or due to this inflammatory response, comes from when you have IL-16, IL-6, and TGF-beta expression, and particularly IL-23. Particularly these guys are high inflammatory. IL-6 and IL-23 have high inflammatory activity. And so when if you have an inflammatory response, and these guys are produced in strong amounts, you will get a favoring of the Th0 cells, which are interacting with the antigen processing cells to transform into these Th17 cells. And as a consequence, these Th17 cells then produce IL-17. I want to point out this TGF-beta, I just have it down here, is transforming growth factor beta. And this is one of the factors that's important in producing the IL-17, the Th17. The goal is to produce the IL-17 and to cause all of these kinds of things to happen in that vicinity. And so these cells don't really kill any cells. They're basically factories for IL-17 and a very high inflammatory response in that area where they are. Now, it's turned out that these are mainly these Th17 cells or Th0 cells you'll find them mainly in the mucosal membranes, and when they are formed due to interactions of Th0 cells and these mucosal membranes with antigen processing cells, then this IL-17 will lead to macrophage activation, neutrophil activation, as I've seen, and chemoattraction. And this will result in an inflammatory response that goes after bacteria and parasites that are within those tissues. However, this inflammatory response can sort of get out of hand, and this is why you need control mechanisms. But in some cases, it can get out of hand and be overly activated, and in some cases, there's association with some bacteria that are in the microbiome, that may be causative of this, as well as other types of where where the immune response is not doing the right thing. It's getting too overinflamed. Uh, these can result in autoimmune disorders such as psoriasis, IBS, inflammatory bowel syndrome, multiple sclerosis, and others. And so this has turned out to be an interesting target. So if you can block the production, if you can block IL-17, in other words, if you can produce an antibody to IL-17 to remove it, then that was investigated as a possible therapy for these autoimmune diseases. And in fact, that has turned out to be the case. For example, this is an anti-IL-17 therapeutic antibody, saxizumab. I never can pronounce these names; are not meant to be easily pronounced. It's produced by Eli Lilly, and it's for the treatment of plaque psoriasis, and this was approved just recently, 2016. And there's a couple other types that have also been in development. There's also, remember, IL-23 is important for the production of the Th17 cells. Go back here. So if we were to block IL-23 here, then we would block the production of Th17 cells and therefore the production of IL-17. And so there's been an antibody that's been Stelara from J&J, which is the treatment of plaque psoriasis and Crohn's disease. And this was approved in 2007. And there's a variety of others that have actually also been under development, have been under development. And so there's sort of two approaches here, blocking IL-17 and removing it, and therefore blocking the effects of IL-17, or blocking IL-23, which blocks the production of IL-17. So those have all been tried, and this is actually for the IL-17, and this is Talzitz. So those have been fruitful, and those have proved to be useful in the treatment of these autoimmune diseases. Now, in the last part of this section, what I'd like to discuss is the regulation of T cell responses. Now, there was a question earlier, and so if T cells get get overactivated, what ends up happening is you can get a tremendous amount of cytokines produced, and it can actually kill you. It's referred to as a cytokine storm, in which you get too much IL-2, too much IL-6, too much IL-17, too much interferon gamma is produced, and this can lead to severe toxicities. So, what's been built into the system is a way to tune the response down. So, in the initial stages, what we've seen is you want to activate the T cell response, and it will go up, and then it's going to sort of plateau, and then there are mechanisms for shutting it down, for starting to tune it down, and to tune it down so it doesn't get out of control, because you don't want to have just an immune response that's continuously getting greater and greater and greater. And so we've talked about here's your antigen processing cell, here's a T cell here, just here, and here's a T cell here. And what we've talked about is the stimulatory effects, right, with CD3 and CD28. Now, it turns out these are positive. There are inhibitory co-signals as well, which block this interaction. And so over time, what happens is there is inhibitory receptor interactions that lead to tuning down or inactivation of T cells, so they stop proliferating, or they proliferate with a lot less efficiency. So it's always a balance between these, sort of going up and then going down within the response. So these molecules that have been developed, have been identified, as it turns out, are molecules in which uh, what's happening here is a substitution. And the most important thing here is this CD. When we looked at the CD28 and the B7, or it's CD80, CD86 interaction. This is a signal that is pro-activation. Now, it became noticeable that there is another molecule that's expressed, excuse me, on antigen processing cells, and that can be produced. This is referred to as CTLA4. And if it gets expressed, as it gets expressed, sure I got this right. Sorry, it's the other way around. As you get an activation of the T cell, what you end up getting is the CD28. You get CTLA4 is being produced by the T cell, as you can see here. And what it does is it's a negative signal. So the CD28 and B7 are up here. They're the positive co-stimulatory. But as this co-stimulatory effect keeps going, you get, as it turns out, as the cells go on, there's a negative co-stimulatory molecule that starts to be produced. And it basically can block this interaction between the antigen processing cell and the T cell. And so that's one way. Another way is the tissues. So we have here the priming, get an activation, and tissues. Tissues can produce a molecule that will also be a negative stimulatory activity. So, for example, normal tissues would produce a molecule called PTL1, and on T cells, antigen experience, so these would be memory-based, or these are not naive cells. These are cells that are that have interacted with the antigen, will produce a molecule called PD1, and the PD1 and PDL1, if they lock horns, if they bind to each other, will produce a negative signal to turn down that T cell. So this is one way that normal tissues have of protecting themselves against T cells that might bind to their MHC1 and they shouldn't be binding to it, or stick too long to them, and to get rid of those T cells. And so it's a way of blocking the interaction between normal tissues and T cells. Now, this regulation turns out to be that tumor cells are somewhat clever in the fact that they, in their oncogenic pathways, when they get turned on, they can produce more PDL1. And what this has the effect of doing is turning off their ability to be recognized by T cells. So normally a T cell that can recognize a tumor cell, for example, because it's recognizing a antigen, basically a mutated protein, piece of a mutated protein, referred to as a neoantigen by MHC, it wants to kill this tumor cell, but the tumor cell has produced PDL1 on its surface because its oncogenes have, and that PDL1 is recognized by TB1 and will basically starts to block this T cell response. Now, so this is sort of part of an innate immune resistance of cancer cells. For adaptive immune response, when you have a tumor cell, the T cell will produce interferon gamma, and the interferon gamma is picked up by the tumor cell, and the tumor cell says, "Hey, I'm being attacked," or the interferon gamma I'm sensing interferon gamma. There's T cells around. I'm going to produce PD-L1. And so they produce, they go through this signal transduction pathway, and they produce PD-L1 on their surface. So sort of, they're ones that are constitutively oncogenic active, produce PD-L1, and then their tumor cells can respond. They don't have, if they're not expressing PD-L1 very much, they can respond by responding to the interferon gamma that the T cell is producing. So, sort of two ways. So these are two ways that tumor cells have of blocking the immune response that would normally that the immune system produces to get rid of malignancies. So and therefore evade basically the immune response, leading to greater tumor formation. Now this set up an idea, and this idea was the brainchild of Dr. Jim Allison at MD Anderson, that if you were to block these molecules, if you were to block this response, then what would happen is instead of blocking the T cell response, in this case for CTLA4, if you were to block CTLA4, then the T cell would be able to undergo continuous T cell activation and also kill the cancer cell. So let me back up here for a second. So if we look here, here's normal T cell activation. There's a co-stimulatory activator. CTLA4 is produced by the T cell, and eventually what happens is these T cells stop undergoing stimulation, and therefore these T cells, which should go after the cancer cells, have been activated, get tuned down because they've been essentially what's referred to as they've become somewhat exhausted in their ability to kill the tumor cells. And so Allison's view, and this was the first case, is that this is one of these negative regulators. And Allison thought that, well, if you had an antibody that bound to CTLA4, then this would allow for CD28 to still bind to B7, and you would still get activation, and that activation would go on to lead to a more powerful anti-cancer immune response. This proved actually to be true. He was actually derided for this idea. When he did this, when he used in this paper, he showed that if you took an animal and implanted a tumor, this is an animal with a full immune system. He implanted a tumor. I believe this was a melanoma-based tumor, and then he gave injections of the anti-CTLA4, either the control or an anti-CD28 or anti-CTLA4. What he found is that if he gave, this is the control, the tumors grew quite well. Of course, anti-CD28 would block activation, so the tumors would grow well. But if he gave anti-CTLA4, the tumors actually, the tumor growth stopped, and it went down to basically zero. Essentially, the T cells of the animal could get rid of that tumor. So this was the first example in which a checkpoint, referred to as a checkpoint, in which if you block the ability of a T cell to be deactivated, you would enhance an immune response to tumors. So his drug, the drug that he developed, this anti-CTLA4, was later developed by Medarex, which was later bought by BMS, Bristol Myers Squibb. This blocks the CD28 from binding from binding to um, uh, CTLA4. That's wrong there. That should be, that's a mistake. So binding to CTLA4. Oh, this is not good. CTLA4. So sorry about that, but it blocks CD28 from binding to CTLA4. It's a humanized mouse monoclonal. It was approved in 2011 for melanoma, and many clinical trials have been used for it, and it has been effective. Its effect seems to correlate with the amount of these. You'll see this in the literature, neoantigens, and these are antigens. These are basically the mutated proteins in the peptides from the mutated proteins in the cancer cells. And it induces a long-term memory effect so that the patient who responds favorably to this is essentially cured of their cancer. And it's particularly good for melanoma because melanoma patients, melanoma has a high incidence due to mutations. It's one of the cancers because exposure to sunlight and all produces a lot of mutations in the DNA, which is derived from, and therefore produces a lot of these neoantigens. And about 1 in 5 have very long-term survivors. Essentially, they're cured. So if you look here, this is ipilimumab with against cancer cells. Against cancer in patients. So here's a lung cancer, and you can see here's the lung cancer, and post-treatment you see a loss of the tumor. Here's another examples in which these little tumors have disappeared. This is a brain melanoma, so I don't know if you remember, you might not remember at all, but Jimmy Carter, former President Jimmy Carter had brain cancer, but what he had was actually not glioma. He had a melanoma that had metastasized into the brain, and that actually was quite, that responded quite well. Now, one of the issues with these kinds of therapies, like ipilimumab, is that you can get side effects, and those side effects have to do with the fact that if you block the ability of the T cells to be regulated by CTLA4, you can affect actually reversible autoimmune diseases in which T cells get overactivated, and you can get dermatitis, colitis, and some hepatitis. These are resolvable. In after treatment, but it points to the fact of the fact that CTLA4 is important. You need to have control and a downregulation of the T cell response, or you're going to get some powerful side effects. Now, this was used clinically. Ipilimumab was used clinically, and this is for melanoma patients. And essentially, after 20, these are metastatic melanoma patients. After 2 years, essentially 9 out of 10 patients have died. And what was really interesting is that when they did this study with ipilimumab, and this GP140 is actually a vaccine that was produced from the melanoma, which we're not going to talk about, but it was done as a comparison. But these sort of like dashes here, this is just the ipilimumab itself. And what you find is that the ipilimumab, that 20% of the patients basically had stable or went into remission. And that was quite remarkable, because usually what happens is in a cancer therapy is, to be honest with you, what happens is you might extend this like this, all right, but it always goes down to eventually down to here. What this showed is that you could maybe, if you got even better, you could maybe you got it, but then maybe the bar goes up here. So now you've got 40%. And so the idea now is can you raise this bar? Because in their first clinical trials, they showed about 20% of patients essentially were cured. And this has been consistent because they've gone out now 10 years. It's been almost 10 years since these patients were treated, and they're still cancer free. And so if we can get better, the idea for this was revolutionary, is that what you want to do is you want to see if you can raise the percentage of people that are long-term survivors, and therefore think about curing more people. Now, obviously, just using ipilimumab isn't the cure-all, because 80% of the patients actually ended up succumbing to the disease. But this gave some hope that actually using this kind of therapy to enhance T cell responses would and therefore enhance the production of basically these anti-tumor T cells, could lead to long-term control of the disease. And this has really taken the whole field of oncology by storm, and is the hottest area right now in anti-cancer therapy. And I want to point out this is 2015, so this is not very long. This is only in the last, you're talking only the last 5 years or more, 5 to 10 years, and particularly in the last, in some cases, some drugs only in the last 4 or 5 years have been approved. So I thought I'd show you, just to give you a taste of Jim Allison for this discovery, for basically founding this whole field of basically checkpoint inhibitors. A couple years ago, won the Nobel Prize for this. And I thought I'd play this little. I thought it was a nice little movie to introduce him, because sometimes we don't think, or we just see the results of the work, but this gives you an introduction to the actual person and researcher who did this work.
It wasn't that I was going out trying to say, oh, I want to find a way to cure cancer. Not at all. That wasn't in my head at all. I wanted to understand how T cells worked.
Without him, without my oncologist, I mean, my kids wouldn't be here. My husband wouldn't have a wife. So it's kind of amazing that because of this one man, that all of that, for me, happened. And I know that I'm just one person, but for me, and for my family, it's life-changing.
Jim Allison's discovery has changed the course of thousands of people's lives, and led to a paradigm shift in how some cancers are treated.
My father desperately, I think, wanted me to be a doctor. He was the old type of country doctor, that did house calls. And so I started college as pre-med, but they had several courses, organic chemistry in particular, where it was really taught in a way to eliminate people who couldn't memorize, regurgitate a lot of facts. And I didn't like that very much. And when I started thinking about it, the responsibility to be a doctor is really pretty incredible. I mean, you need to be right. When you're a scientist, you're supposed to be wrong. And you can be wrong all the time, so long as you're right every now and then. What I've learned since then, if you're not wrong most of the time, you're probably not out there like you ought to be.
In college, Allison got interested in T cells.
There's really cells that go around your body and can detect virus infection or cancer or even things your body's never seen before, had never existed in the universe before.
When they detect an invader, T cells go from scouts into warriors. And what Allison discovered, which would be so useful for treating cancer, is that the T cells don't just turn on, they also turn off by way of a protein called CTLA4.
You got to stop T cells because they'll go from a few hundred to tens of millions in several days. You can't let that go on.
Allison wondered if this natural braking system prevented our immune system from defeating cancer. He thought, what if you could disable the brakes long enough to give the T cells a shot against a tumor?
Just take CTLA-4 out of the picture, and then you can let the T cells go as long as you want. Well, that was the basis for the idea.
The idea was not particularly well received.
A lot of people had just given up on the idea that you could use the immune system to catch, because there have been so many failures. Even in immunology, some of my friends that were very renowned, they wanted to insult me in public. They'd say, "Jim's a tumor immunologist," snigger.
It took years to convince a pharmaceutical company to develop the drug. But when the clinical trial results came out, they were dramatic.
Moved the median survival over by 4 months.
No other drug had moved median survival at all. But even more promising, long-term survival improved. After 4 years, over 20% of patients treated with the drug were alive. None in the control group had survived.
That had really never happened before.
It's insane is what it is. I mean, I had tumors in my lungs, in my brain at one point.
Sharon Belvin was diagnosed with cancer just before she was to get married. She was 22. She tried chemo and radiation. Nothing had worked. That's when her oncologist, Jed Walchuk, started her in a clinical trial for Allison's drug.
Over the next couple of months, my tumors kept getting smaller and smaller and smaller until my doctor finally said that there was no more evidence of disease.
I was in my office, and I got a call from a clinician I work with, Jed Walchuk, and he called me and said, "Jim, come over to the outpatient clinic." I said, "Hey, man, I'm busy." I said, "Just come over here."
Dr. Walchuk says, "Well, the guy that kind of invented this whole thing is here."
So she grabbed me, hugged me, and squeezed me.
I said, "I'm sorry, but I just got to hug you."
Everybody started crying.
You saved my life.
I got an email from her, the big photo of her first baby. And then a couple of years later, her second baby.
There's no words to describe what it feels like. Oh, I'm trying not to get all emotional here. What it feels like when you have handed someone back their life.
Yep. I wish it worked more than 22%. So I was a basic scientist, but I still think you can't be so pure that you don't think about what benefits may come to people from your science. And if you could do your basic science and then sit back every now and then and say, I wonder if this could do something, then you ought to do that.
Okay, so that gives you a little taste of some of the impact of these kinds of therapies. Now, this has gone on and has led to explosion into looking at other types of checkpoint inhibitors, because it turns out that there are other ways, there are other molecules that will also control T cell interactions with tissues, not just CTLA-4. One of those molecules is PD1. And this is another one in which is controlled as an immune checkpoint, in which if you block the antibody, if you use an anti-PD1 inhibitor, you can then induce activation of the T cells to express activation of the T cells to go after and to kill the tumor cells, enhances tumor infiltration. So the recognition of PD1 and PD is actually the fellow, the professor is Japanese. I can't remember his name right at the moment, but he won the second part of Jim Allison's Nobel Prize that was given to Allison for CTLA-4 and to the discoverer of PD1. Anyway, this led to the development of now potentially thinking of anti-PD1 based inhibitors to do a very similar effect. And it's turned out that that's been a very powerful response as well, such that if you use an anti-PD1, and now this is a drug nivolumab. Here's a patient treated with renal cancer. Here's tumors before treatment, and you can see they're gone, basically. Whole tumor masses have been eliminated. Here's a patient with melanoma. Here's the melanoma down here, this patient, and you can see after treatment, the melanoma has been resolved. He's had these terrible lesions here. Look at his ear, and these have undergone resolution. And you can see here a patient before and after, massive tumor amount of tumor here has been nearly completely resolved. This is the power of the immune system when it's activated. It's absolutely stunning what it can do. It's unlike any sort of chemotherapy response. It can just melt tumors, basically. Patients with non-small lung cancer have been particularly useful for. This is an interesting case because in this case, here's before treatment, here's the lesion, the lung, non-small cell lung cancer, and actually at 2 months, it's actually gotten bigger. Before in 4 months, it's been completely resolved. And you can see that down here too. Here's another lesion down here. Let's see here, it's gotten bigger and then resolves. What it turns out is that they found out is that when you biopsy this, these tumors are actually so infiltrated with T cells that they actually get, the whole mass gets just bigger. And so this actually is a positive. When they initially saw this was like, oh my, this is not working. But let's keep going and let's see. And luckily they did keep going. But it turns out that what's happening here is that the T cell, this is actually a positive response because it's actually the T cells are actually getting activated, getting in there, and are filling the tumor T cells and causing, and then this thing resolves the lesion. So nivolumab is also from BMS. It's an anti-PD-1. It blocks that interaction from PD-1 and PD-L1 on the tumors. So PD-L1 is produced by the tumors. If that wasn't clear, I just want to make sure that is. Go back. So that PD-L1 here, which interacts with PD-1, is blocked. The black interaction with PD-1 and PD-L1 is blocked. And it's a humanized antibody, was approved just 5 years ago, and as many clinical trials are underway with it and other types of for other types of cancer. Effectiveness does not have a consistent correlation with the amount, interesting, of PD-L1 expression on the tumor cells, which is interesting. And neoantigens are important for some cancers, but not others. So it's turned out that there's not a complete correlation between the ability to express on the MHC1, the mutated peptides from the mutated antigen, mutated proteins. And it induces a memory effect, although this memory effect has turned out to be not as long-term as might have been expected. As long-term as, for example, the CTLA4. There's reasons for that, and I won't go into that right now. But if you ask me in class, I can discuss that. Another one is pembrolizumab, which it blocks also. It's by Merck. PDL blocks the interaction for PD-1 and PD-L1. Another humanized PD-1 monoclonal. And this was approved in 2014 for advanced melanoma after treatment with ipilimumab. So the CTLA4 treatment isn't really working as well, and so they add in a pembro. It's been approved for non-small cell lung cancer and patients that express PD-L1. So they and failed chemotherapy. So they want to know it's most effective against patients who express the ligand PD-L1, which is the inhibitory ligand in the cancer cells, which makes sense. And this does seem to correlate with expression of PD-L1. And in the FDA approval, actually, patients you only give this to patients that show that they express PD-L1. Otherwise, it would be pointless to do that because it's not as effective against those who don't. And you can see this expression, PD-L1 expression in non-small cell lung carcinomas. There's just there are different levels in different. The point of this slide is just to show you that depending on the type of the expression levels, the cancer cells from the patient, what you see is that there are different expression levels. The dark color is a staining for PD-L1, and some lung cancers express small amounts while others are loaded up with it. And so it turns out that it's most effective against ones that have a high expression. And so that can be, you monitor that. Those are the patients that would be most likely should be treated. Now it's turned out to have a very positive effect. So this is, you can see here the bar has been raised quite a bit with these patients, with the number of patients who've been, what this shows you is that patients that have expressions of PD-L1 do better than ones that don't. And so this expression level is, they have at least greater than 50%. Then you see 60% survival actually for 3 years versus 30%. So it does matter. And you see again, this is what we're trying to look for: raising this bar of so that you this is actually the survival curves. So it's a survival number of people who survive is much longer. So remember, PD-L1 and PD-1 interact with each other, and this will block T cell activation. Whereas the peptide, MHC1 peptide, T cell receptor activation, and leads to activation. And just so you're aware, what's left out here is CD3 and CD28. It's just been stripped down to make it simpler. So this whole interaction leads to stimulatory interaction, whereas PD-L1 and PD-1 lead to negative feedback on the T cells. So the idea is, could you also block PD-L1, is not just have antibodies that bind to PD-1, but also to PD-L1, which is on the tumor. And so, so the rationale is anti-PD-1 blocks interactions with PD-1 on the T cells with PD-L1, and there's another binding molecule, PD-L2. It's not as necessarily as important. And then anti-PD-L1 blocks interactions of PD-L1 on the tumor cells with the T cells, but not PD-L2. And so you can do either one. You can bind either here or you can block here. Those are the two possibilities. So a drug has been tested. So antibodies to anti-PD-L1 antibodies have been developed, and you can see here this is before treatment in a melanoma, and before treatment and then after treatment, 3 months to 10 months. You get 3 months. You notice the tumors actually get bigger, and then they get smaller. And so you're getting activation, they're starting to fill out with T cells, and then you're getting destruction and removal of that tumor. So quite an effective response. You can see here in non-small cell lung cancers, in which you have these tumor masses before treatment and then after treatment. You can see these tumors have been completely resolved from this patient. You see complete resolution of those tumors. So Atezolizumab is an anti-PD-1, a PD-L1 antibody. So this binds to the PD-L1 on the tumor cells. This again blocks this PD-1, PD-L1 signaling. This was approved just 4 years ago now, 3 to 4 years ago, for lung cancer, bladder cancer, triple negative breast cancer, all in the last 4 years, and particularly after treatment. In some cases, after treatment with ipilimumab. For some cancers, the treatment is after chemotherapy failure and or inability to take chemotherapy. So this has proved to be also a useful modality for either blocking again the PD-1 on the interaction by having an antibody that blocks to the PD-1 on the T cell, or an antibody that binds to the PD-L1 on the tumor cell. So both have been tried, and there's multiple efforts to try to develop even new and better ones ongoing right now, and in a variety of combinations, emphasizing the fact that when you do that, when you block these basically down regulatory responses, what you get is a enhanced immune response, and therefore a higher anti-cancer response, a better anti-cancer response. I will say the major side effect is from these kinds of therapies is some loss of control of T cell behavior, and therefore, as an effect, you get basically these induced, potentially induced short-term inflammatory responses or autoimmune responses, in which T cells that are overactivated start to cause problems with normal tissues. In most cases, those can be resolved after the antibody is gone. In some cases, it has proved has not been resolved in a small number of patients, and so that's a major issue to try to see about how we can remove that. So with that, I'm going to stop and and if you have any questions on Monday, let me know.